Good morning, everybody. Oh, you so fast. You beat me to it. All right, give people a moment to roll in before we start. If anyone has questions in the meantime, please feel free to ask. I'm going to gather my thoughts. Mm-mm. Do, do, do. I always wonder what people who are out in the larger YouTube universe think when they see one of my videos on YouTube. Probably doesn't pop up very often, but it'd still be interesting. Oh, there's some guy on his couch not saying anything to anybody, and then they just go and watch something else. Okay, so maybe it helps. I don't know. It's like therapeutic. It's like, those pictures of like burning logs or something you can put on your TV. So it feels like you're warm. Um, I don't know. Anyway, so what I want to do today is finish up linkage. We have a little bit of buffer in the schedule, so we have time to finish that last 3-point cross that was on the PowerPoint. And so again, the reason why I'm getting you to do so many of these things is because practice does really make perfect, right? It makes life a lot easier. And so what I want to do is work through this with you all, right? Now, I'm obviously not going to tell you the answer. I actually don't even know the answer. I haven't done this one. But we can work through it using Dr. Matt's patented 3-point cross method. Okay, so I'm just going to ask you a bunch of questions and then you're going to tell me the answers, right? Or you're going to kind of work stuff out for me because you all have fancy calculators and stuff and I don't. All right, so what's the very first step in a 3-point cross? What is the very first thing you do? Well, actually, it's kind of one or two things, but they're kind of related. Doesn't really matter which way around. What do you need to do? Yes, both of those, right? So, as Sophia said, you need to look for the fewest progeny because those, as Jimena said, if I pronounce that right, are the double recombinants, right? So that's one thing. The second thing is find the parentals, right? Which are the offspring that resulted from the parental chromosomes carrying through, right? So of these, and we need both of those, right? We need both of those to figure out gene order, right? Because we need the parentals because that's what the chromosomes look like originally, right? And we need the double crossovers or the fewest progeny because we need to use those to figure out gene order. Okay, so which are which? What are the parentals? What is their classic characteristic? Right, it's the largest numbers, right? So we have, so let me type this out. I'll use the whiteboard in a second too. They're wild type and all mutant. Right, which ones are the double crossovers? Yeah, but which ones are those? What are their phenotypes? Spineless and scarlet ebony. Okay, right. So let's write this out. Get that over there. Right, let's go find my pen. So this is, what is this? ST, E, and SS. Okay, so we know that the parents are, I'm just going to write them with pluses because I find that just a little bit easier. Plus, plus, plus. And I'm just going to write it as it is on here, which is SESS. Okay. Now we need to look at what chromosomes, what recombinant chromosomes do the double recombinants have, right? So if you look at the up here, right, all of these have a triple mutant chromosome, right, underneath them, right? So effectively, that doesn't actually matter. What we care about is the one that's on the top, right? Which in the case of wild type has plus, plus, plus. Case of mutant is STESS, right? So what is the chromosome of, oh, actually, here you go. Let's ask in a different way. Which allele, which gene moved to create a spineless or a scarlet ebony? Which one is moving? So if you look here, it's SS, right? Excellent, Jimena. Because in spineless, we have plus, plus, and then a recessive, right? In the scarlet ebony, we have recessive, recessive, plus, right? And so the one that is different from all of those three, because in the wild type, they're all the same, is SS, right? So this is wrong. Actually, I didn't need to rub that one out. It is ST, SS, and E, right? This is the correct gene order. Because the only way that we can have just the S moving alone, so this goes over here, or this goes over here, is if SS is in the middle. Everybody got that? Awesome. That makes me very happy. Okay, and then the rest is pretty straightforward. All we have to do now is figure out what things are moving, and then we can figure out what are the single recombinants. All right, so let's figure out what's the single recombinant between ST and SS, right? And so if we have a single crossover event here, we're going to end up with ST plus plus and, oh, actually I should do it over here. ST plus plus and plus SSE, right? Because only this one is moving, right? It's basically going over here, and this one is going over here. And so we need to look and find which ones are basically spineless and ebony, and which ones are, what's the other? Scarlet. There you go. So we need to find, oops, oh yeah, I did spell that right. And spineless and ebony. Right, so what numbers, which ones have those phenotype combinations? What are the numbers for those? If you look through that kind of list of phenotypes, which ones have that? spineless and ebony, and scarlet. What are the numbers that I need? It's going to be 50 and 52, right? So spineless and ebony is 50, and this is 52. Excellent. Yep. Azucena, almost, not quite. Hang on. Nope, Jimena, you're off the beaten track. Spineless and ebony, and then just scarlet alone. Have a look again at that list, Jimena. Hopefully that should make sense now. Right. Okay, so are these the only numbers that we need to add together to work out recombination frequency and distance between scarlet and spineless? Is it just these two added together over the total? Yeah, that's it. Plus the double recombinants, because double recombinants still have recombination occurring between these two, right? So we have to add those, right? So the distance between these two is 52 plus 50, right? These two, plus the double recombinants, which are 5 and 3, right? Which gives us, what's that, 102, 110? The total down at the bottom of that picture is 755, so you don't have to add it up yourself. Now, if someone can work out, let's tip this down a little bit more. If someone can work that out for me in terms of percent, what is that? And then it's times 100, obviously. Awesome, 14.6%. I'll be careful not to round up, because those numbers after decimal place are actually important, right? Because if you're comparing distance. Okay, I'm gonna take a picture of that. Right, keep that in mind. Yeah, Jelly, I don't think we need quite that many decimal places. Like super accurate. All right. Now what we want to work out is the other one. So crossover between these two. And so that will get us plus E and STSS plus. Right, so that should be ebony and scarlet and spineless. All right, what are the numbers for those? Ebony alone, 43. And what about scarlet and spineless? Yeah, 41. That's actually probably why you're typing, right? And so let me just hang on a second. This is such a clunky thing. Now, Jimena, you can see why when you put 43 and 41 before, you're a little bit off base because you put down scarlet, but it's actually scarlet and spineless. And we really need to be very careful about making sure we're looking at the right things. And so that's why when you try and work these out, writing out the gene order, the wild type and the mutant actually makes life a little bit easier because you can literally just put a cross between the ones you're looking at, and then that will tell you if this is going over here, these are the phenotypes and the phenotype in this case that you need to care about to work out this distance. Right, so this is the most, it's a little bit more laborious, right? But it's the most straightforward way of figuring this out. No, I'd always, if I want you to work out a 3-point cross, I have to give you the numbers, because otherwise you'd actually have to do the experiment. And I think that would be a little bit unfair in an exam, but it'd be kind of interesting. But oh, good, Jimena, I'm glad, that makes sense. Yeah, Amber, yeah, the only way that you can work these out is with it if you're given all of the numbers, right? So this isn't really a situation where I would give you part of the information and expect you to work out the rest. Essentially, the rest will be the genetic distances, that is, and the gene order. Those are the two things you have to work out from this. So yeah, it is really quite straightforward. You know, and that's the cool thing about this, is that once you understand what you're doing and you follow the, you know, Dr. Matt's painted in 3-point cross plan, it's really straightforward, right? And so the trick to doing well at the exam with these kinds of questions is just doing them a bunch of times until it becomes second nature and easy, right? Once it gets easy and you're like, yeah, I can just do that, give me a couple of minutes and I'll work it out, then you know that you've got it. Okay, so what other numbers do we need to work out recombination distance? It's exactly the same as before. Yeah, okay. And it sounds a little bit like I'm kind of banging away on the same drum, which I am. But the reason for that is that if you forget these numbers, you'll get the wrong answer. And given that these are so important, hint, hint, one of the answers to a question like this may include the calculation without these numbers in it. That answer would be incorrect. I'm not trying to trick you. I'm just pointing out the fact that these are really important and you have to include them. Okay, so that's a little bit of a kind of a heads up kind of deal. All right, so that is what, 94 divided by 755 times 100 equals, dang, you fast, 12.2%. So now all we have to do is put those into a map and we're done. So it's going to be, did I have that up wrong? I think I got that right. 94. 84 plus, oh, shit, I'm bloody useless. Actually, we might need to work the other one out now. Yes, Sophia, you're 100% correct. Oops. This is why I should use a calculator. What's the number now? Sorry. Was it still 12.2? Yeah, awesome. Okay, sorry about that. All right. Dang it, now I'm gonna have to take another picture and remember to delete that old one. Did I do the other one right? Let me just double check. Yeah, I did do the other one right. Ah, isn't that crazy? How can you do one thing right and then do the other one wrong? It's exactly the same numbers. All right, so now it's simply a case of draw this out. We have ST, SS, and E, and this one is 12.2 centimorgan. This one is, I think, was it 14 point? Oh, I've got to scroll back up. This isn't very user-friendly. Yeah, 14.6. And that's it. Right? Nothing else to it than that. So if you follow that plan and you just make sure you're not making any leaps or, in my case, bad mental arithmetic, then you will always get it right. It's really that straightforward. There's nothing else to it than that. So any kind of question I give you like this, the answers that I'm after are gene order, distances. And that's it. Anybody got any questions? It is. I mean, the funny thing is probably when you started this, you're like, oh my gosh, this is just awful, right? Crazy complicated, don't know what I'm doing. And then you do it and it's like, oh, it's actually not really that hard. You know, once you understand what's going on and follow the plan. Okay, so just to finish up, a couple of clarifications, right? So we have different map types. What you've been working out are linkage maps, right? Or recombination maps. These are based on the frequency of recombination between different loci. However, we also have actual physical maps, right? These are literally the number of bases between different genes, right? So if you look up fly base in yellow, all right, this is the recombination map here, which basically says yellow is right at the very, very end of the X chromosome, which is called 1 in Drosophila terms for some reason. But its actual physical location is this. These are the actual number of bases. And it's on the long arm of the chromosome, right? If you're to look up something else, for example, all right, this is recombination group 36. So this is way distant, right? And the actual numbers, I think the other ones were like 3 million and something, right? Now we're up to nearly 12 million bases, right? So now you can say, oh, those are really far apart, but you can both say they're this far apart in terms of linkage and this far apart physically, right? And generally speaking, by and large, those two are the same. So I just got, oh yeah, so good point, Amber. When you look at something like this, it looks wildly confusing, right? There's all kinds of stuff going on. That's why you need to break it down into those kind of very deliberate steps. Because if you just look at that as it is, it's like, what's going on? It's just a whole load of gibberish. But when you break it down into those kind of little bite-sized chunks, it's really quite straightforward. So getting back to linkage versus physical maps, The reason why they're not, and typically they do correlate really, really well, particularly as I think I've mentioned before, in the bits of the chromosomes between, like the arms, between the telomeres at one end and the centromere in the middle, right? Recombination basically ties with distance, physical distance. I'll do have a calculator. Where this doesn't pan out is either on the Y chromosome in humans, because there's very, very little recombination there because it's so teeny short, and it doesn't match the X chromosome very well. And also in the centromere, right? So the region towards the center of the chromosome, which is typically silenced, right? It's typically heterochromatin. You don't get a direct correlation anymore because there's so little recombination here, right? And so essentially this is kind of like a dead zone with regards to recombination. And so things will appear much closer together by linkage than they are actually by physical distance. Genes just either side of this dead zone. And that's pretty much that. Anybody have any questions before we move on to the next one? Awesome. Oh, okay. Yeah. Feel like spring break should be about here, but unfortunately it isn't. Okay. Let me just get rid of this in case I need it again. Yeah, I know. It kind of makes you wish you had both a longer Christmas break and spring break, but unfortunately you can't get both. So it's going to be a little bit tough kind of the last half of the semester when it's like, also for anyone that has kids as well. To be honest, that's going to be also tricky. Okay, so what I want to do now is kind of roll back a little bit. Need my coffee. And touch back again on stuff that we covered a little bit earlier, right? So here's that kind of reiteration, right? This going back over stuff to make sure it really sinks in well. And what we want to talk about today, or start talking about at least, is we need to get going through this whole bit about DNA replication, homologous recombination, and so on, right? And so that will lead up to, get out, exam 2, which is going to be not this Friday, but the Friday after. I don't know what it is about small flies, why they need to fly right in front of your face. Anyway, so we've done our whole linkage part. This week, we're going to dive into the bulk of DNA replication, right? And that's largely going to be split between prokaryotic and eukaryotic DNA replication. And prokaryote is a little bit more straightforward, a little bit. So we typically cover that first. And then eukaryotic DNA replication will come after. And then finally, we'll end up going through the actual mechanism there of homologous recombination, right? How do we get those crossover events? What's the molecular basis of that, right? We're seeing the inheritance outcome, and we're actually looking at the numbers and how that ties into physical distance, right? But in this lecture on the probably on the 17th, maybe 15th if we make good progress, we will look into actually how does that work. So what's here? Oh yeah, that was a snow week, wasn't it? Yeah, so Sophia, correct. Exam 2 will be on all of the non-Mendelian patterns, which range from dominance, epistasis, linkage, through DNA structure, DNA replication, and homologous recombination. That will be what exam 2 will be on. So it's going to be a bit of a wide range in topics, right? Because we're going to be going from very much probability and math-based stuff to very much molecular type processes, where you'll need to remember the names of things, a lot of the names of stuff, how things work, right? Whereas this is a lot more kind of like higher level, what are the outcomes of these different things happening, right? And then we're going to start getting into the actual molecular basis of that. Oh yeah. I'm sorry, I need to have coffee. Should have had one before class. Oh, we have another exam on that day? Ah, that sucks. So yeah, just try and plan to do one in the morning and one in the afternoon, I guess. Would be the deal. And I will also be doing the same deal where I'll actually sign into the Zoom at 9 o'clock in the morning and hang around for a bit, see if anybody needs any help with the exam. Pardon me. Okay, so all right, so essentially this is a picture that isn't necessarily going to make a whole bunch of sense right now, but it should make a bunch of sense by the time we get to the end of this topic. It's actually kind of a neat, doesn't have everything in there. Yeah, it's kind of neat. I like that. Anyway, so we've got to go right back to the basics, right? And the basics being building blocks of DNA and how they're put together, right? We know that we have purines, which are 2-ring nitrogenous bases, pyrimidines, which are single ring. Actually, it's kind of handy. These letters in red are the electropositive or electronegative regions which allow hydrogen bonds to form, right? So here you can see we have 2. We know thymines bond with adenines. Adenines also have 2. And it has one slightly negative, one slightly positive, one slightly negative, one slightly positive. That's why thymines and adenines pair so well. We also have a sugar and a base, right? So if you just have a sugar and a base, it's called a nucleoside. When you add a phosphate group on, it becomes a nucleotide, right? This is obviously DNA because, as is helpfully pointed out, we only have one hydroxyl group, right? If you were to have 2, one on the second carbon, it would be an RNA molecule. Okay, and these properties are going to become really, really important when we think about DNA replication. The main ones being this OH down here, right? And also the phosphate group to a large extent, or 3 phosphates if you have a trinucleotide. And also the basis of pairing, right? These groups here, these electropositive and electronegative regions are, and also the size as well, right? So 2 goes with 1, oops, this one. This 2 goes with this one, right? So you only have a maximum of 3 kind of rings between the backbones. These 2 properties are what determine a large part of the accuracy of DNA replication. Largely because a guanine will not easily bind to a thymine. It kind of will-ish, but not very stably and not as neatly. Similarly, adenine really isn't going to do a good job of binding to either another adenine or a cytosine or whatever, right? So the exact formation of hydrogen bonds, because we've got a negative, what's that? Plus, negative, negative, and this is plus, plus, negative, right? So we have the exact opposite. That is how we get a lot of the accuracy in DNA replication. Recap as well, this 3' OH is so important because this is where new nucleotides are added, right? You cannot add DNA or RNA bases to the 5' end of a strand, right? This actually causes issues in eukaryotes much later in terms of their linear chromosomes. Pretty big issues, actually. And that ties into all kinds of things as to the reason why you get old, which sucks, and the reason why some cells can escape that and become cancerous, which also sucks. There's not a lot of joy in this, right? We need to kind of like, yay, happiness and stuff. But most of the times we talk about things, it's about stuff that goes wrong. Right. Anyway, and so the big take home from this is that DNA and RNA and protein, right, for the same reason, which we'll get into when we look into gene expression, have directionality, right? They are different from one end to the other. And the big deal in both DNA and RNA is that you can only add new bases to the 3' end, right? So keep that in mind for when we talk about transcription. It's exactly the same principle applies. And also just for notation basis, the bonds that hold these bases together are called phosphodiester bonds. I often call these bases, right, because that's kind of like a hangover from terminology, because these are called base pairs, which is a unit of distance, right? Gene or length. Gene is 3,000 base pairs long. The actual base part of the nucleotide is this part here. It's the nitrogenous base. So that's a little bit of kind of notational kind of flexibility. Five to three Hennessy?
That's something my chemistry professor would say.
Oh. Anyway, so one of the issues then becomes the fact that this is a anti-parallel double helix, right? Which causes issues if we go down here, is we have two strands, right? We've got this one strand here, 5' goes all the way down here to 3'. We have another strand which goes, sorry, I'm getting distracted by noise from the kitchen. Where is it? 5' all the way up here to 3'. Hey, peeps. Evie. Don't forget I'm teaching, okay? Just a little distracting, that's all. Okay, so these strands run in opposite directions, like right so. But because DNA replication can only proceed in the 5 to 3' direction, that means that we're going to have two strands being synthesized in opposite directions, right? For the 5 to 3', well, this one, right, going down, the new strand has to be synthesized going up that way. The other one, the 5 to 3, which just goes up, now the new strand has to be synthesized going down. This will cause all kinds of issues later on, particularly with bacterial and with eukaryotic DNA replication. Okay, we've already covered the hydrogen bonding. Actually, one important part that comes out of this is a simple numbers game, right? So you know that G's and C's are held together by 3 hydrogen bonds, A's and T's with 2, right? Now, which of these collection of bonds, either G's and C's or A's and T's, are going to be held together more strongly? Between which 2 bases will the bond overall be stronger? Between G's and C's or between A's and T's? Right, between G's and C's, because they have 3. So if you need to open up DNA, yeah, that's right, Jimena. If you need to open up DNA, what would you expect to find more of? G's and C's or A's and T's? If you need to periodically open up that stretch of DNA, right, A's and T's, because it takes a lot less force to do so. And so if you look at origins of replication, right, which is really wherever in a DNA molecule DNA replication begins, or in transcription where copying DNA into mRNA begins, you will find stretches of A's and T's. Right? In bacterial transcription, you have something called the Tata box, because you largely have T's and A's in it, right? If you look at any origin of replication, there will be a stretch of A's and T's near to that, which is where when you twist DNA, it will pop open, right? And so this isn't just important for accuracy of DNA replication. This bonding between these 2 bases is also really important for the actual physical properties of DNA, right? Okay, another interesting kind of aside, tying to like the big scale stuff. If you're looking at a whole genome of a bacterium, right, and you found that bacterium came from somewhere astonishingly cold, right, really, really, really cold. It would be called a psychrophile, which is kind of neat. What would you expect to find more of in its genome? A's and T's or G's and C's? Remember, as things get colder, you have less energy to do stuff with. Biochemical reactions proceed more slowly. So which of these would require less energy to break apart? A's and T's, right? So when you look at the environment, right, of an organism, that is also expressed in its molecular makeup, right? So if you look at hyperthermophiles, organisms that live at 110, 120 degrees Celsius, right? This is actually above the boiling point of water at sea level, right? And they manage that because they're below sea level, right? So the pressure is high, right? Those have a predominantly higher GC ratio than the other ones. Exactly. They need stronger bonds so their DNA literally just doesn't fall apart, right? So those bonds aren't broken just by the energy in their environment, right? And so that's one thing that I try and do throughout this course is a lot of this stuff isn't just abstract. It also ties into like the very big picture of ecology and evolution as well, right? It really is what underlies everything. As I've often said, until I start talking to a chemist and then they trump me with their bloody chemistry as basis of everything stuff. Anyway, mutter, mutter, mutter. So this is important. This is not so important, but it's also neat to know, right? It's a right-handed helix, again, anti-parallel. This is also astonishingly stable. What is it I read the other day? They had sequenced DNA from, I don't know, woolly mammoths again or something, which was something like 2 million years old. Yeah, it must have been from like within its bones or something, because there wouldn't be much else left, right? But this is astonishingly stable molecule, right? It hangs around for a very long time. And I've got a little aside. Has anybody heard of Ötzi, the Iceman? I think that's how it's spelled. It's actually really neat. I don't know what I was talking about. Maybe I haven't talked about Death Valley and stuff in Antarctica. But anyway, is an individual that was frozen in a glacier, right? And in Austria, right? Glacier, way up in the Alps. And he was spat out a little while ago when the glacier retreated and just basically left him on the snow. And he was killed about 4,500 years ago, 4,000 years ago. And they actually figured out, they think he was shot in the back with an arrow. So he was running away from someone who didn't like him very much, who killed him with an arrow. Isn't that crazy? And they could figure out what he was eating, what his diet was, what his general health was based on, kind of things like that. I think that's super, super cool. But you can do all kinds of really crazy stuff with DNA simply because it hangs around for so long. Proteins don't, generally speaking, because they get used as a food source by microorganisms. Same with carbohydrates, which are much less interesting. RNA doesn't because it's extraordinarily fragile. But DNA will hang around for a very, very long time. So that whole kind of Jurassic Park deal of getting DNA out of insects stuck in amber, it wasn't really all that far-fetched. It was based on actual science. The whole making dinosaurs from that was science fiction. But anyway, yeah, that's right. Yeah, the whole ability to kind of create dinosaurs is a little bit tricky. But you'd start with birds, right? They are the closest thing that we have to dinosaurs, essentially, or one kind of offshoot. That and crocodiles. Okay, so just to finish up, one thing that is, let's just move this over here so I can see what I'm pointing out. One thing that is very important about DNA is not just its actual bases, right? The information that is contained within it, also its shape matters, right? So DNA is not an equal twist. It's not like you get two strands of string and just kind of, or wire and just like twist them up. So you get an equal kind of twist around them. It's a kind of that kind of twist, right? Where you have the two strands together and you have an uneven twist, right? Where you have what's called a minor groove and a major groove. Now, this is important because there's an awful lot of proteins that recognize DNA, but they also recognize the shape of DNA, right? And so they'll actually physically interact with the DNA and fit into those grooves, right? And so what we'll talk about a lot later on, particularly in control of gene expression, is DNA binding proteins, right? Proteins that will recognize stretches of DNA and cause a response in one way or another once they do. Okay, so just to kind of finish up the last couple of minutes, just a couple of things to talk about. One is what I'll be doing with all of these kind of molecular processes is breaking them down into phases, right? Everything that really pretty much that we have begins with initiation, right? You have to start the process. You have to find the region that you want to do something to, in this case, start copying it. In the case of DNA replication and actually transcription, you have to unwind the DNA, right? So don't forget, all the information is on the inside, right? We need to kind of open that up so that those nitrogenous bases are now accessible. And in the case of DNA replication, we're going to use those as the template to make the new strands, right? Each one. In transcription, we're just going to use one of them as a template to make RNA, which then disappears and these things go back together again, right? In DNA replication, we open stuff up and then we're going to basically turn, this one is going to become one new DNA molecule, right? Consistent of the old template and the new one. This one will become the other new DNA molecule, right? So essentially, when you go from having your two strands, this is one chromosome, opens up. After replication, you'll have your two chromatids, each of those with two strands of DNA. Right. Obviously, you have to elongate the new DNA strand, right? And then you have to finish things up, right? And there's a lot of stuff that goes into this. So it's worth kind of keeping in mind like the big picture, what's going on and why is it going on, right? Why do we need to do the things in termination that we do? Why do we need to do the things that we do in initiation, right? Try and keep those in mind, and then it becomes a little bit less of an alphabet soup, right? The actual purpose is what matters. The names kind of come with that. All right, I think that's enough for today. Let you get on with the rest of your days, and I'm going to make myself a cup of coffee because I'm not from talking to you lot, just from being pie-eyed, and it's Monday, I guess. Yeah. All right. Cool. All right, everybody. Shall see you soon. Take care and have a fun rest of your day.